So, good morning. So, generation and conduction of the nerve impulse. How the nerve impulse generated and conducted throughout the body parts. You know that different types of neuron helps in our transmission of the message from one part to another parts of the body. Our brain is also helps in this transfer or generation of the messages. How this is occurs. Neurons are the excitable cells because these membrane are in the polarized state. Neurons, they are excitable cells. Some injury, if we get some mosquito bites, we get some anything touches our body, our skin, we can give the sensitivity or sensation and that reaches to our brain. Okay, then brain give the advice and that reaches to the different parts of the body through this neuron. Okay, they are too much excited, their sensitivity, if any type of really small type of touches occurs in our body, the neuron get the sense. Neurons are excitable cells because their membranes are in the polarized state. Polarized state means some ions are present there. Do you know why the membrane of a neuron is polarized? Why the membranes is polarized? Because this is the channels that permeable to the ions, that is selectively permeable to the different ions. Okay, selectively permeable means that is allow entry of some specific ions, not all ions can enter, all ions can goes outside. Selectively that is useful that, and some there is some pores are present that is particularly for specific for the some typical. Some pores allow entry only Ca plus, some pores allow entry only Na plus. Selectively permeable means if they are allowed the Na plus, only Na plus can goes outside and inside, and none other can goes inside the cells. Okay. When a neuron is not conducting any impulse, the resting stage, obviously when they does not work, when they do not work any type of the work, it is in the resting stage. Conduction means the impulse is transmitted by the neurons, that is not occurs. When a neuron is not conducting any impulse, resting axonal membrane comparatively more permeable to potassium ion and nearly impermeable to the negative charge protein present in the exoplasm. Okay. So, when at the resting stage it can allow entry the K plus ion and impermeable to the sodium ion. Okay. This is not allowed in the resting stage. When a K plus enters inside the neuron's body, then goes to the resting stage. And exoplasm, the cytoplasm of the axons also present, that is also not allow the entry of the any type of negative ions. Okay. If K plus is also negative, that is not also entered through the exoplasm. Okay. That is why it is selectively permeable membrane. Similarly, the membrane is impermeable to the negatively charged protein present in the exoplasm. Consequently, the exoplasm inside the axon contain high concentration of K plus, exoplasm inside the axon K plus higher concentration and negatively charged protein and low concentration of the Na plus. In the contrast, the fluid outside the axon contains low concentration of the K plus and higher concentration of Na plus and thus form a concentration gradients. Obviously, K plus enters through the exoplasm, so that is why inside the cell K plus is the higher concentration and Na plus does not enter inside the cell. So, that is why the outside of the body there is the page number 317, Vishal, 317, generation and conduction of the nerve impulse. So, you know that the osmosis diffusion always the ion or any type of things occurs goes inside the cell from higher concentration to the lower concentration region. Here resting stage, so it allows only K plus ion enters inside the cell. So, K plus ion concentration is higher inside the cell and Na plus just goes outside the cell. And concentration gradients means there is difference, there is some difference present, so that is why pressure is present. Na? So, then Na plus goes inside the cell, K plus goes outside the cell due to this concentration gradients. Resting membranes are membrane by the these ionic gradient across the resting membrane are maintained by the active transport of ion by the sodium potassium pump, which transport 3 Na plus towards to Ka plus into the cells. Okay. So, due to this concentration gradients, Na plus, if 3 Na plus goes outside the cell, 2 K plus goes inside the cell. Okay. This is the 3:2 concentration gradients of the sodium and potassium ion. As a result, the outer surface of the axonal membrane possesses a positive charge, while its inner surface becomes negatively charged. Okay? So, axonal membrane outside positively charged, inside negatively charged. Therefore, it is polarized, and the exoplasm inside negatively charged, negative always destructive. So, that is why any type of negative ion cannot get inside the exoplasm. Okay? Only positive ion get inside the exoplasm, because the inside is the negatively charged and outside is the positively charged. The electrical potential differences across the resting plasma membrane, and this is called as the resting potential. Potential means capability. They can enter, they are capable to enter this type of ions, and this is occurs at the resting stage. So, that is called resting potential. Resting potential K plus inside cell is higher, Na plus outwards is higher. When the Axon potential started, Na plus is lower the outside, Na plus go inside, K plus outside. Okay? Just opposite. You might be curious to know about the mechanism of generation of nerve impulse and its conduction along the axon. So, how the obviously we are eagerly waiting to know how the concentration gradient create and it transfer the messages from one neuron to another neuron. One stimulus is applied at a site on the polarized membrane. The membrane at the site A becomes freely permeable to the Na+. So, one side of the axon, that is a receptor stimulus that helps to stimulate, activate. Something must be present that helps to activate that site. Na? So, there is some site A that is in the axon, that there is some stimulus is present that helps to activate. Stimulus means some gate injury, some sensation that stimulates the axon. This leads to the rapid influx of Na plus followed by the reversal of polarity at that side. So, at this time Na plus got inside, K plus outside. The outer surface of the membrane become negatively charged, and membrane is also in the resting stage, the outer surface positively charged, inner surface negatively charged, but in the axon potential outer surface negatively charged, inner side positively charged. Becomes negatively charged and the inner side becomes positively charged. The polarity of the membrane, the site A, is thus reversed and this depolarized. Obviously, reversing occurs, positive outside, inside, negative outside, depolarized. The polarity of membrane site A thus reversed and then depolarized. The electrical potential difference across the plasma membrane at the site A is called the Axon potential. Okay? The polarity of the membrane at the site A thus reversed and hence depolarized. The electrical potential differences across the plasma membrane at site A is called Axon potential. So, now differences occurs at the site A. So, that is known as the Axon potential because they are activated, which is fact as term as the nerve impulse. So, this active potential is also known as the nerve impulse. At the site immediately ahead, the axon membrane has a positive charge on the outer surface and negative charge on the inner surface. As a result, the current flows on the inner surface from the site A to site B. On the outer surface, current flows from site B to site A to complete the circuit of current flow. Okay? So, just opposite site A to site B, then B to site A, that is the current flow occurs. Hence, the polarity at the site reverse and Axon potential generated at site B. So, first Axon potential started to site A, then it polarity reverse, then negative positive opposite, then it goes to the site B. Thus, the impulse Axon gated site A arrives at the site B. The sequence is repeated along with the length of the axon and consequently the impulse is conjuncted. The rise in the stimulus induced permeability to Na plus is extremely short lived, that is quickly followed by rise in permeability to the K plus. So, you know that axon is the long like hair like structure. So, one site activated, another site is deactivated, then that is reversed to the site A, then it goes to step by step site A to site B. First Axon potential occurs site A, then it reverses to the or goes to the site B. Okay? And Na plus stability is very short. So, that is why then again K plus enter inside the cell, then it goes to the resting stage. So, that is why this is the repeated, the first resting stage, then active stage, then resting stage. This is the nerve impulse conduction. It is quickly followed by a rise permeability K plus within a fraction of second. K plus diffuses the membrane and restores the resting potential to the membrane at the site of excitation, and the fiber becomes once more receptive to the further stimulation. Transmission of impulses. How the impulse is transmitted throughout the body. A nerve impulse is transmitted from one neuron to another neuron through the, so first action potential, resting potential occurs within one neuron. Now this transmission how transmitted to the another neuron, okay, that is transmission, and that is occurs by a junction one neuron to another neuron, that is called synapses, okay. A synapses is formed by the membrane of a presynaptic neuron and postsynaptic neuron, which may or may not be separated by a gap, is called synaptic cleft, okay. So two neurons are present just side by side and they form the synapses, okay. Gap between, there is a gap between the neuron. This like the structure is, there is the axons are present and there is also another axon is present and that is formed. So like head like structure, so there is the axons is present. So synaptic cleft, maybe here is formed, maybe dendron is formed, and there is the junction that is known as the synapses, okay. That's there may be gap is present, may not be gap is present, that is known as the synaptic cleft. There are two types of synapses, okay, namely the electrical synapses and chemical synapses. So some synapses occurs through the electrical conduction. Electrical means here is the ion, okay, positively charged and negatively charged ion, and the synaptic classes, and chemical synapses, some hormones, maybe some things that helps in their transmission. So at the electrical synapses, the membrane of the pre and post synaptic neurons are very close proximity. Electrical current can flow directly from one neuron to the another neuron, these synapses. Transmission of one impulse across the electrical synapses is very similar to the impulse conduction along a single axon. Impulse transmission across an electrical synapses is always faster than the across a chemical synapses, okay. But electrical synapses is the faster than the chemical synapses. Electrical synapses are rare in our system. Our body functions, that is grows slowly, so there is the chemical hormones, different types of chemicals are present in our body, biochemical structures, protein, lipid functions. So our body contains the chemical synapses. At a chemical synapses, the membrane of pre and post synaptic neurons are separated by a fluid-fed case, that is called synaptic cleft, that is known as the synaptic cleft. How no presynaptic neuron transmitter? So how the presynaptic neuron transmitted the transmission to the post? Pre means previous, post means next, after that, okay. Pre to post, how it is transmitted? It is transmitted, chemicals is called neuron transmitter. So the chemicals that helps in the transmission, that is called neurotransmitter, that is related to the neuron, that is called neuro, that helps in the transmission, that is called transmitter. So neurotransmitter helps in the transmission or conduction of the nerve impulse from one neuron to another neuron. The axon terminal contain vesicle fields, that is neurotransmitter. When an impulse arrives the axon terminal, it stimulates the movements of the synaptic vesicle towards the membrane where they fuse with the plasma membrane and release their neurotransmitter, the synaptic cleft, okay. Some stimuli that is joined with this synaptic cleft and helps in their neurotransmitter secretion. The released neurotransmitter binds to their specific receptor. Here is the neurotransmitter release and their specific receptor is present, that if Na plus is present, that is there is the specific transmitter, there is some receptor is present. Receptor means helps in the receipts, okay. Present on the postsynaptic membrane, the binding opens ion channels allowing the entry of the ions which can generate a new potential in the postsynaptic neuron. The new potential developed may be either excitatory or inhibitory. The brain is the central information processing organ of our body and acts as the command and control system. It controls the voluntary movement, balance of the body functions and vital involuntary organs. So central nervous system, our brain is our central nervous system, because the brain control all the functions, all the emotions of our body. So neuron give the messages to our brain, then brain tells us what should we do and what not, and according to the brain advices we do our work, that is control center of our body. So that is why brain is the called central nervous, main nervous system. Brain is the central information processing organ of our body and act as the command and control system. It controls the voluntary movement, balance of the body, functioning of vital involuntary organs, lungs, heart, kidneys, etc., thermoregulatory hungers and thus circadian rhythms of our body, activates of several endocrine glands and human behavior. It is also the site for processing of vision, hearing, speech, memory, intelligence, emotions and thoughts. The human brain is always protected by the skull. Our brain is always protected by the skull, the heart structure. Inside the skull, the brain is covered by the cranial meninges. So, the covering of the brain is known as the cranial meninges, and it consists of outer layer dura mater, a very thin middle layer arachnoid and inner layer pia mater. So, our brain covering meninges has 3 layers: dura mater, arachnoid layer and pia mater. Dura mater outside, arachnoid middle, pia mater inside. That is the layer of the brain covering, that is known as the meninges that present beneath the skull. Okay. Next, the brain can be divided into 3 major parts: forebrain, midbrain and hindbrain. Our brain totally divided into 3 parts, that is forebrain, that is front part, the hindbrain, that is the backward, and the midbrain, middle part. Okay. Now, forebrain. What is the structure of the forebrain? Cerebrum. Forebrain has 3 parts. It consists of 3 parts: cerebrum, thalamus and hypothalamus. Okay. Our brain has 3 parts. Forebrain has 3 parts, that is hypothalamus, it's called cerebrum, thalamus and hypothalamus. Cerebrum is the main part. So, forebrain consists of cerebrum, thalamus and hypothalamus. Cerebrum forms the major part of the human brain. A deep cleft divides the cerebrum longitudinally into the 2 parts. Okay. So, cerebrum divided the human brain into the 2 parts, that is by a deep cleft or deep pit, drain-like structure that divided the cerebrum into the 2 parts and known as the right and left cerebral hemisphere. Okay. And these 2 hemisphere is jointed by the nerve fibers, that is known as the corpus callosum. The forebrain consists of cerebrum, thalamus and hypothalamus. The cerebrum forms the major part of the human brain. A deep cleft divides the cerebrum longitudinally into the 2 halves, which are termed as the left and right cerebral hemisphere. The hemisphere are connected by a tract of nerve fibers called as the corpus callosum. The layer of cells which covers the cerebral hemisphere known as the cerebral cortex and is thrown into the prominent folds. The cerebral cortex is referred to as the grey matter due to the greyish appearance. Okay. So, cerebral cortex covered the cerebral hemisphere and it is the covering of cerebral hemisphere and it is known as the grey matter. They are grey in color. The neuron cell bodies are concentrated here giving the color. So, why it is grey in color? The neuron cell body concentrated, gathering from the colonies, and cells, they are cell mass, they are grey in color. So, that is why our grey matter is grey in color. The cerebral cortex contain motor areas, sensory areas and large region rather than the clearly sensory nor motor in function. This region called as the association areas are responsible for complex function like intensensory association, memory, communication. So, memory is present in our cerebrum, cerebral hemisphere, that is cerebral cortex. Okay. So, what is the function of the cerebrum, what is the function of the forebrain, all are same. Okay. Memory is present, hungerness, eating, drinking, emotions, all are controlled by our forebrain. So, the neural cell bodies are concentrated giving the color. The cerebral cortex contains motor areas, sensory areas and large region. They are the neither clear sensory motor in function. This region is called as the association areas are responsible for complex function like intensory association, memory and communication. Fibers of the tracts are covered by the myelin sheath which constitute the inner part of the cerebral hemisphere. They give an opaque white appearance. Now, cerebral hemisphere, the inner part is white in appearance, so that is called white matter. So, white matter and grey matter, outside grey matter, inside white matter. According to the center of sensory and motor signaling, another very important part of brain is called hypothalamus, lies at the base of the body temperature, ergo of eating and drinking. It also contains several groups of neurosecretory cells which secrete hormones called hypothalamic hormone. The inner parts of the cerebral hemisphere and a group of associated deep structure is amygdala, hippocampus, etc. with the hypothalamus. It is involved in the regulation of sexual behavior, expression of emotional reaction, excitement, pleasure, rage and the motivation. Okay. So, this is the function of the forebrain. So, this is up to for the today. We will continue the rest of the part in my next video. Okay. Thank you very much everyone. If you have any question, you can ask me. Bye everyone.